we get experimental science. We're curious, non-judgmental.
So why this is a cool study. Synthetic silk is something that people have been after for a long freaking time. That is because it is one of the most powerful complex molecules in terms of how much strength is there. The idea of what they've been wanting to do, and this is in the journal Nature Communications, so it's a pretty high tier publication, a synthetic spider silk. So spider silk is considered to be very strong and ecologically friendly. Why it is famous? Its strength, its flexibility, it's lightweight, it has a really strong tensile strength equivalent to steel, and strength to weight ratio that there's nothing like it in the world. So from a building perspective, from a sustainability perspective, everyone's trying like, let's make spider silk. So in this study, what they found was they created a device that can spin spider silk. So if y'all have actually been watching us play Grounded, in Grounded there is a device that you can build that allows you to spin spider silk. Turns out that it does not end up happening in the game as in real life, right? You can't spin spider silk on a loom made out of twigs and berries and leaves. Instead, it is this extremely complicated microfluidics chamber. Here it is. It's this custom chamber that they built, and here is the little dish right here. And what ends up happening inside this dish is that it recreates the molecular structure of your spider silk via chemical and physical changes that normally are occurring in the spider silk gland. So the spider silk gland is undergoing a bunch of reactions, and there's also motions of pulling within the gland that create this silk. And so they had to figure out a way to replicate that in a machine, which had never been done before. So the spider silk fibers, if we were to do the really high res zoom in images, there are beta sheets in there, and it must be aligned properly. So if they are at all misaligned, instead of being stacked on top of each other like this, these sheets, if there's a misalignment, all of those really amazing properties of the silk, like the tensile strength and all the forces behind it, it just falls apart. And that's been like a really big struggle in science, is getting all the sheets to align the appropriate way so you can replicate. It's not just that you're making the same molecules of the silk, right? It's that getting it the same features and characteristics, like a true synthetic spider silk that has all the properties of it, both physical and molecular. And so this allowed them to do it. So this device, it looks like a rectangular box. They use protein sequences from a specific spider species. But yeah, they were looking at like what the molecular characteristics are of spider silk. In this chamber that they built here, this microfluidic device, what they're doing here is that there is this precursor solution that's put here in slot 3. And then pulled towards this end. And this is actually apparently the really big push of it, that it's not being emitted through that other side of the rectangle via any kind of strong force. So in the past, what they have done is like syringe pushing, that they ended up like pulling the sample out from. And apparently that kind of force was changing the beta sheets from being stacked on top of each other to having these misalignments, and that ruins the strength of the synthetic spider silk. So they wanted it to be stacked on top of each other, so they actually use negative force, so it's naturally coming out of this end of the machine, or of this plate here, which is really, really cool. It's kind of like if you have a really thin glass tube, like a capillary tube, and you stick it in some water, the water will naturally try to go up the tube against the force of gravity. So you're essentially replicating that within this microfluidics chamber, where the fluid is naturally being pulled out one direction. And that prevents the shearing from happening, keeps all the sheets together. Yep, but exactly, very, very thin. And it's naturally being pulled out. And that was, I guess, the big kicker as to why this ended up not having before. Because they said when it wasn't these correct conditions, the proteins just kind of fell apart or were loosey-goosey. But with all these correct ones, like the right concentrations of liquid, the right pHs, they had to figure out like what pH was ideal, and then this negative pull force. With that collectively, they were able to replicate the spider silk. See, that's the issue. Like, making the silks generated by bacteria is really cool, but it doesn't have the physical forces that you need to give the properties of the silk of what they should end up being like, which is what that microfluidics chamber was needed for. The tactical underwear, listen, GK, it's really cool though, right? Like, of all the different things that this spider silk can be used for. Like, it's not limited to just like buildings, but like just I don't know. I didn't think about the possibility of using it for protection in clothing, and it's just really cool. Like the unlocking the secret of how to make spider silk, like in a lab, could be a game changer for a lot of different fields and areas of, you know, not just of research, but like production. And I think that's really remarkable that something that exists. So the interesting part about this, when they were talking about the different kinds of clothing, the synthetic spider silk coming from bacteria, that could be used for regular clothing. Like, it's a silk that you can make, yes, and you can make clothing out of it. That won't have those unique physical properties that make the silk so powerful, you know, for, like we talked about, the buildings, like the tactical underwear is not coming from your bacterial made silks. It's like how it gets produced. That is the big component of it. Like, it needs to have undergo this particular pressures and changes. Goes to produce spider milk. For some reason, the story did not inspire me to say that should work. So OG, the reason, like looking back on it now, is because in the microfluidics chamber, you could argue that the different pressures that you need might be replicated in the mammary gland of the animal. I mean, it's a stretch, but knowing the data from this paper, I can kind of see where they're going from it, right? Like, you're putting in the genes to synthesize the same molecules, right? And that will create this, like, at least the composition of the silk, which is pretty remarkable. So it was like this combination of extra features that they made on this microfluidics chamber that hadn't been identified as important pieces in the past. And now they have it down, and so they can actually make that synthetic silk. So yeah, it was a combination of things that they identified in the study, and their hope is that they can then use this synthetic silk for a lot of different factors. How do spiders do it? So the spirouettes that they have, OG, apply force in particular ways. And so the issue is we don't understand how the spirouette applies the force and where it does in the process of making the silk. And so if it's like some flavor of negative force inside the spirouette, or like what else is going on, like they don't know enough about that. It's more of a black box with the spirouette. Now, what exactly still happens to the spirouette, we don't know. I wouldn't be convinced until they have some really hardcore genetics in there as well. So you'd have to have tissue specific drivers, meaning tissue specific genetic removals in the spider, without which you wouldn't be able to really nail down what's going on in the spirouette. And they just don't have that kind of genetic resolution at this stage. You can, you know, adjust and change things on the fly, but I'd say you still need those before you really know what exactly is going on. Maybe one model is, OG, that there is some negative pressure going on in addition to some kinds of physical force on the silk, but how all those things work together is still unknown. Not that people aren't studying it, but it's neat that the synthetic now has a means by which you can get it to work. This video outlines why we can't make spider silk 5 years ago, and it's kind of neat to think about in the context of, well, why can we then make it today? What about today allows it to make us to make it? And so the answer to that is us looking at the paper again and that microfluidics chamber and what they ended up doing in creating that scenario to be able to have synthetic spider silk. I think there is a lot of physics behind it, and some of it is absolutely over my head because of like the physics behind it, but the way it was explained to me, and it was explained in the paper, was essentially if you're taking a capillary tube, so just like a thin glass tube, and you put it in water, the pressure, the water rises up, so it's going against gravity. And so somehow they've engineered that phenomena to be able to have the machine push out the silk in that manner. And it has to do with, it's like a negative water pressure that they're building in. So it's, again, a water-based solution. And so it's like going against the rate of flow, and that's what fed into being able to have this work. I know it's not an ideal definition, Ratfinky, but it makes a smidge of sense in my head. Let me know if it makes sense at all. Yeah, Hogan, they had this paper come out like 2 weeks ago where they are finally able to make synthetic spider silk. And the big catch of this is that you have the beta sheets that make up the silk strands, instead of being off each other, they are now aligning and they're parallel. And because of that parallel structure, the tensile strength is maintained. And that's what allows them to actually use this for like future, like, again, like skyscrapers or buildings or whatever. Like, that's like the idea of it. Like, yes, is that a future stretch right now? Maybe, but it's much closer to reality than ever before, which I think is really remarkable. I think it is pretty big bug news, Bean. Yeah. So their big limiting step, chat, in using this microfluidics chamber right now is, well, now they're able to get the tensile strength right. So the spider silk is as strong as it should be. But the catch now is, as Sparky points out too, is that the production amount still hasn't increased sufficiently. So the amount of spider silk that they can put out now that has the correct tensile strength is not yet where it needs to be. So it's not like we can just get a pile of spider silk that's like at least correct functionally from this microfluidics chamber. They're going to have to upscale this, and that could, be affected by costs and a bunch of other different distinct factors. But it's getting really close, and hopefully we'll be able to use this technology for building many a thing. It was neat that they had some really pretty imaging as well of the tensile forces and like what the spider silk looked like. They even imaged real spider silk versus the synthetic spider silk that they worked on. So you got a little bit of everything throughout this paper. Again, y'all know I'm a sucker for imagery. They did like 3D reconstruction of the spider silk as well, along with the synthetic. You can see here in this figure, D&E, how all of those lines are moving in the same direction. And that's, again, those beta sheets that are aligned parallel. That was the key trick to be able to get from this study. Do they know how long this would last if they make it? So they don't know how long it would last yet, Sparky. They did mention, like, they're able to cause it to break down, so it is biologically able to fall apart. And they did demonstrate that, but it was not under the conditions where it would be representative of what would be going on in the world, right? Of like how many times it can actually survive this kind of thing, or if there's extra treatments that they put on. They didn't do any of that. It was just like a very intense way of pushing it through the process and seeing, okay, what's happened. So just they wanted to demonstrate it does biodegrade and not demonstrate how long before it biodegrades. But you're right, Sparky. I guess their assumption was that because it's super close to spider silk, it will last a fair bit of time. But yes, they need to go through and characterize that as well. I think upscaling too is really high on the list. If they can't upscale, then this is all dead in the water. They demonstrated they did have the beta sheets again, using the microfluidics chamber as well as what a real spider does. They simulated the shear stress that the fluidics chamber can do. The current best way. So the current best way, OG, that you can produce the spider silk, and this is what's publicly available, OG. There are some companies that do this. I have no idea what their methodology is, right? Because it's all behind proprietary stuff. In terms of amount, it's still probably like getting microorganisms like E. coli to produce the silk to be able to have it. But that's just producing the material. There's nothing right now that is publicly available, not behind, some kind of trademark or whatnot, that has the same tensile strength. It's tougher than steel, Matt. Tougher than steel. And in both like tensile strength, in the physical strength of what it can hold, like it is a really, really amazing molecule. But a lot of that isn't replicated in the other means of producing the silk. So you need to be able to have, like, that's why this microfluidics chamber is so important, is because it captures all the properties of it that we want. Like, yes, you can make, like, of the not so strong variety, you can make a shirt out of it, but it's not very efficient in terms of production right now, and then it's also like not capturing the properties that you would want for it that would make it any better than having like the silk from the silkworm. And yeah, there's the cross section of what the fibers look like, and they also then like write out how they find that these spider silks assemble within the spirochetes, and that's what they based the microfluidics chamber on as well. So they're trying to base it on existing biology in terms of what ions they're using, what solutions they're using. It's still not quite on point, and we don't know the genetics behind it, but some of the physical forces we might know more about. And that is this paper. So that is our look into the silk assembly with the microfluidics.